Hello, everyone. Thank you for staying with us until the end. I really appreciate it. Cha Vaccine Institute is utilizing leading uh, immunotherapy technologies to be a leader in biotechnology industry, and I'm going to talk about what immunotechnology we have, what um, infectious disease we're trying to target and cure, as well as anti-cancer drug development plans as well. So Cha Vaccine Institute is connecting industry, academy, research, and hospital. It is a Cha hospital connection and infrastructure, and we are considered a hospital. We have a bio-focused infrastructure, and we are focusing on how we can effectively develop our own products, and our environment is very favorable. Our main headquarters is in Seongnam, and the research center is in Pangyo. And as I shared earlier, we have a immunity building platform, and we are applying this technology to many different areas. Actually, we are focusing on premium vaccine and vaccines needed in the aging society, as well as anti-cancer vaccines and treatments. So now, to briefly introduce our company, we have TLR23. This is TLR targeting, especially targeting 2 and 3 elements, and we have been focusing on infectious diseases and anti-cancer vaccines as well. So when we are identifying pipelines, we look at unmet needs and the chance of success, and whether we are globally viable and capable or not before we choose a specific area of research. So to introduce our platform first, we have an adjuvant platform technology, and adjuvant is basically is enhancing or adjusting vaccine reaction. It's a immune related material and ingredient. Even though it's not the main ingredient, it serves as a very important element. And if you look at the left or right side, when you just put the antigen, the immune response is quite weak. But if you put the immune builder, which is adjuvant, you can see that many different immunocells are gathered together around the T cell, and there is activation, there's interaction, there is split of activated cells, and then B cell is activated again, so that cell response or antibody reaction can be enhanced as a result. That's the role of adjuvants, I would say. So what's marked in blue over here is specific roles of adjuvant. Currently, in the pandemic situation, I wanted to emphasize how important of a role this adjuvant can play. First, it can quickly increase the protection level. So if you're using a great quality adjuvant, immunity can be built, and you can also use less antigen. So that means you can distribute more number of vaccines, and because immunity is going to be built, not just your immunity is improved, duration will last longer as well. So the effect of vaccine can last longer, and especially for a situation where mutants and variants keep popping up, you can expand the impact of the vaccine as well. And because it is targeting new T cells, it can increase cell response immunity so that it can effectively prevent the severity of symptoms and preventing a patient from suffering from severe symptoms. And what's problematic is that vaccine reaction is quite short, and mutants are popping up, and mutants do not get tackled by existing vaccines. But if you include and introduce adjuvants, which is immunity building, you can actively overcome the existing problems, and as a result, this lead to the better usage and impact of vaccine effect itself. And when the craze was going on, we were not able to quickly respond to this use of adjuvant. That's one unfortunate thing. And there is aluminum um, ingredient, and it alum or non-TLR, and this slows down the overall process. So you can see that Toll-like receptor is there as a feature. And this is working as a immunity booster. So Toll-like, which responds to TLR ligand, and then immunity reaction is garnered, and then this intrinsic immunity is connected to non-intrinsic or environmental immunity responses, and we are focusing on the role of TLR targets. So when you say TLR, there are about 13 different existence in immunocells, and in our own company, what's special about our adjuvant is that TLR2, which is membrane number 2, and intracell TLR3 over here. You can see it over here in the cell. And these two types of TLRs are targeted specifically, and these ligands are combination ligands, and they work in different locations to generate synergetic effects. To be more specific, on the left side, there's L-PAMPO. This is the complex of TLR2 and TLR3 ligands. And as I mentioned earlier, there is a great synergetic impact that we can achieve. And there are two receptor targeting mechanism to adjust immunity as well. So if you look at L-PAMPO, this is for chronic hepatitis B therapeutic vaccine. Even though it's a new adjuvant, we were able to verify its safety. And the right one is LipoPAM. So we are usually encapsulating the material, but now what we're doing is putting this onto the liposome, so that liposome itself can carry immune boosting features. And this is why small-scale antigens can be delivered, and mRNAs can be delivered utilizing the system. So when we use this one, cell immunity reaction was boosted. It was an immunity builder, and we are using for recombinant herpes zoster vaccine as well. And now I would like to talk about how our adjuvants are better than other options. So left one is showing how L-PAMPO is increasing cytokine as much as possible, and the right side is COVID vaccine animal testing results. If you look at the left corner, there is famous adjuvants and TLR ligands, and if you compare different ingredients. On the top side, that's a singular independent use. On the bottom side, when they mix up and combine different ingredients, you can see that whether it's independent or mixed use, our L-PAMPO is showing the greatest potency. And you can see that these are adjuvants that are used in regular COVID vaccine development process. First indicator is neutralizing antibody, and the bottom graph is an IFN, LA spot. And if you look at our L-PAMPO trademarks, you can see that whether it's antibody or LA spot, we are showing a great impact and efficacy in both indications. And when we carried out an indication therapy or testing, our immunity boosting was proven, and this was posted on vaccine-related science journal as well. This is quite a complicated table, but we implied many different target vaccines compared to existing adjuvant. When we use our independent adjuvant, how is it going to be better? You can see that we are encouraging and inducing larger areas of immune responses, and even for the vaccine options that are being developed, you can see the efficacy was derived by using our adjuvant. What I marked over here is Herpes Zoster vaccine and HPV vaccines, and these are going through clinical trials. And the first line over here is GSK's technology. I would say GSK is the player which is the biggest and making most amount of money around the world. And I can say that this is a company that has a great and profound vaccine portfolio. There are adjuvant series such as AS4, 3, 1, 15, and 2, and I would say representative material is MPL and QS21. That's the representative adjuvant systems, and of course there are more as well. And at Cha Vaccine Institute, we have L-Pampo, we have Lipopam, and other adjuvant systems so that we can expand more platforms to target other indications as well. So these are the types of adjuvants we have, and I'm going to give you some examples for vaccine use cases. So for hepatitis B prophylactic vaccine, chronic hepatitis B is basically is maintained, and there are a number of patients. The market size is quite big, and you have to cure this for sure because it could develop into a liver cancer. There's a risk factor. As a result, you have to treat hepatitis B. Currently, there is antivirus treatment, and the market size is quite big. But current option, you have to take regularly. Of course, we can prevent the virus from spreading or increasing in number, but you cannot remove it. You cannot eliminate the virus itself. Once you take it, you have to keep taking it. And during the process, of course, there is resistance that's built up, and sometimes the medicine doesn't work in some cases. That is why many companies are trying to face this challenge and overcome the challenge, and many are focusing now on boosting immunity. So when hepatitis B, acute ones, they tend to fight everything, but for chronic patients, it seems that they're not going away, but it should be able to be gone as well. So by taking antivirus and by boosting immunity, let's do the combination therapy to fight the disease. So that is why we're now developing a therapeutic vaccine. And of course, just using preventive vaccine to cure the disease, we have seen many different failures. So we are not going for this particular option. You can see that modified vaccine antigen, and the right side is showing a graph. You can see that doing 2 doses of injection, antibody level is growing significantly, unlike the existing one, whereas antibody level is quite low and the timeline is quite short as well. So adjuvant, we use L-Pampo, and the antigen we use modified 3rd generation AG, which is LHBsAg. And in order to develop this therapeutic vaccine for hepatitis B, we focused on whether we can generate immune responses, cell responses, and we used mouse testing. This is mimicking patients' status and circumstances. The first graph shows whether it was going up or not. If you look at the right side, there's no immunoresponses at all, right? But if you look at the second graph, when you put our adjuvant with the antigen, immunoresponses are being induced. And we also looked at whether it is converting to Th1 or Th2. You can see that Th1 response is derived. And if you look at the very right graph, among 7 animals, the 7, in 5 animals, all the particles, antigens, were totally gone. So it's not only removing the antigen, it is boosting immunity at the same time. So based on this result, we went for clinical trials, and we targeted 53 different patients we used a new immunity builder, we wanted to check how frequent, how much we can inject in individual patients, and we set specific dosing, and a 1-2 phase is showing, which is pretty remarkable. T cell activation was recorded in a number of patients, and we saw that there is a potential, and we are now going through the 2B process. So I think the injection is going to be done by this year, and the total closure is going to be next year. And on top of this clinical, we are currently working on licensing out process as well. And there are many antivirus developers at the moment. They want to overcome existing challenges and they would like to develop other indications. But the problem here again is during the injection process, the impact is good, but when the medicine is not given, at any moment, there will be a rebound. And we are targeting to terminate the disease. So, okay, then using the same content, we wanted to see what kind of impact we can expect from a preventive perspective. Let's say using 001 program. Let's say you get a preventive vaccine and you can't get antigen. What can you do? Oh, you saw different clinical studies. 5 to 15% of people are non-responders, and no matter how many times they get vaccines, antigen level doesn't go up, and you can't implement a protection layer for them. And we saw a potential of using this to boost immunity for 001, and we're now going through 002 clinical trial at Bundang Cha Hospital. Once the injection is done this year, we'll be able to wrap up the study next year, and I'm excited to share the result in the future. And now let me move on to the recombinant herpes zoster vaccine.
So here is a specific trend here. So usually you get herpes zoster when you get older, but usually there's VZB when it also happens in early childhood. And even if you catch the contract the virus, it stays latent in your human body. And when your immunity decreases, especially when you get older, and even among young people who get exposed to extreme stress, then also they get herpes zoster as well. But what matters is chronic pain. So when accompanied by chronic pain, then there's actually no answer to that. So it's really important to prevent herpes zoster. And there are two big categories. The alternative vaccine on the right is you weaken the live virus and inject that in the body. And the recently approved SK and Merck vaccine are two types. On the right is the innovative vaccine, GSK, under the name of single Rix, went into the market, and as you can see, it was a game changer in the market because it was so effective. So when you take a look at the left bottom, the blue one is the Shingrix vaccine, and the gray one is the weakened poisonous one, Zostervex, and we are working on the blue category. So this GSK vaccine is so effective. However, it's very painful, and it might cause body pain. That is because of immuno booster, so because of a specific particle, and we use alpha and po. And during the clinical trial, there was no such side effect, so we are expecting that it won't cause side effects for herpes as well. If successfully done, then there will be no side effects. And we compared the two. The yellow one is the commercial one, and the blue one is ours. So there's a huge gap of course, the blue one has much higher efficacy. So in the US, we purchased single X to compare this. And here, the yellow one is the commercial one, and blue one is ours. So the second clinical trial turned out good. So without pain, higher efficacy was the result. So we will keep working on it, and we currently applied for it. And within this year, I think we'll keep moving on to other clinical stages. Next is flu vaccine and COVID vaccine. So I'll move on to the combo vaccine. So for a long period of time, we have been working on this vaccine combo. As you can see, the gray bars here are the conventional vaccines. The higher graph is those young mice, and the low graph is for elderly mice. So in Korea, we offer vaccine for free for the elderly in the national level, and when we inject them beforehand, then the vaccination efficacy recovers. And there's room to calculate the efficacy in the elderly age group, and the called A and B virus efficacy was significantly improved. So we can expand the vaccine efficacy based on the bad flu data. And here are other data of other companies' vaccine flu. And the very right one is our company's. And we compared at the early stage of injection and afterwards. And the sky blue ones are similar, but if it goes pink, then it decreases. But our adjuvant showed a very continuous efficacy. So based on this result, our ALP-AMPO has improved vaccination efficacy, and it has more expandability. We can move on to vaccine A and B with continuous efficacy. And as I have mentioned, here are COVID-19's results. So there is immunity efficacy. So based on considering both results, like those bad flu virus or the COVID pandemic, so our adjuvant, this will be a very good candidate for this adjuvant vaccine. So let's come up with a combo vaccine. If we have to get injected this on a routinely on a regular basis, then it wouldn't make sense to get vaccinated thrice or four times every year. So no matter how well we can respond to COVID, but coming up with the new vaccine every single time might not be so efficient. So if we want to successfully prepare for an endemic, it will be efficient to come up with this based on our adjuvant. So work on bad flu vaccine and COVID vaccine. So we began working on inventing a combo vaccine. So this is under the project of Advanced Propelling Project. So once you contract influenza, then there is scientific basis that it's highly likely that you will contract COVID as well. So no matter if it's bad flu or COVID, the elderly group, they are at risk. They're very vulnerable against these vaccines. So that's why I briefly talked about COVID, and now I'll move on to infectious diseases. So as expanding pipeline, we're working on anti-cancer. So if you take a look at the very right part, dark green color, I would say. So these three circles are what we are working on, are the vaccine and our own anti-cancer. We are working on them. So the L-PAMPO, the immunity booster, can they be used here as well? Well, they're vaccine adjuvants. So of course you can prevent virus, and the cell used for anti-cancer or tumor microenvironment. Under that environment, do they have a pro and anti-cancer characteristics? How well does it balance between the two? So we conducted some research, and we're currently working on writing a research paper. So if I would show you a result, well, so I only have 3 minutes left for my presentation. So this data shows the result. So the uh, suppression was really well done. So the efficacy, the immune system survived at the end. So Aston used our material to conduct a comparative research, and it showed good tumor growth suppression. So Aston is working on a specific antigen, and we conducted licensing out, and we are planning to do a co-research. And this shows some specific data, which was announced last year at SITC 2021. So this is the last point that I would like to say. So there was an interesting point here. So it has immunobooster efficacy, and also it has cancer killing traits. So it kills cancer tumor cells, and when we injected this directly to tumor cells, and it killed and got rid of tumor, and it increased immunity. So immune cells gathered here. So it showed vaccination effect. So that is shown in the middle image. So the cold tumor turned a hot tumor. So because of that, other immune system also became adequate. So the right picture shows thanks to the inhibitor as known, it increases the efficacy by 30% for the treatment. So our adjuvant, our independent efficacy, and the combo efficacy, we are working on both. So this is our own platform. It shows high efficacy, and when used in a combo platform, so the highest result is the control and APD and LMPO. So when combined, it showed 90%. Okay, I have 2 slides left. So there was this combo effect, and when CD8 T cell, when used together, the infiltration rate was really high. And also, this paper was went public at ESMO Congress 2021, and it confirmed that there was good efficacy. So we have good adjuvant. So I would like to say that we should collaboratively work to achieve good projects. So our CHA Vaccine Institute, under the motto of better health, better life, and better future. So all the members can mutually develop toward the future. Thank you.